Hi everyone, welcome back to another video. So this is going to be photosynthesis practice and it goes with the photosynthesis video that was published yesterday. So photosynthesis, very important to know. You'll see that there's relatively more questions about it on USABO. Also very important to know just for general like AP Bio, any bio classes you might have, Science Bowl, Science Olympiad, all that type of stuff. So pause the video and take a look at this one. It comes from the 2017 USABO Opens exam. Okay, so it's asking for which comparison is incorrect. So let's just go through all of them. The first one says Rubisco performs the initial carbon fixation step in C3 plants, whereas pepase or pep carboxylase performs the initial carbon fixation step in C4 plants. So this is correct, as in C3 plants you go directly into the Calvin cycle in your mesophyll cells, and then for C4 plants you're going to essentially fix your carbon dioxide to PEP with pepase. So yeah, let's look at B. C3 plants lose water and energy through photorespiration. That's correct. While C4 plants have no or very low photorespiration rate. That is correct as well. That is basically the improvement of C4 plants over C3 plants. C, RuBP initially accepts carbon dioxide. That is correct. And C4, it's PEP initially accepts carbon dioxide. That's correct. So C is good. D, most plants are C3 plants, and tropical grasses. Most C4 plants are tropical grasses. So D is correct. So we have E. Let's look at it anyways. Spatial separation of carbon fixation and photosynthesis. That's not really true, because it's all happening in the same chloroplast. So for C4 plants, it says temporal separation of carbon fixation and photosynthesis. That's not correct. Those are CAM plants. And then the description for C3 plants, that matches C4 plants. So yeah, then we know that the answer is E. All right, same thing, pause the video and see if you can do this one. Okay, so it's got a pretty long description, but you really don't end up needing any of it to answer the questions as long as you remember kind of the early experiments that were done with photosynthesis. So if you look through all of these, you'll find that D is correct. Oxygen is formed from the splitting of water during photosynthesis. You split it into your protons, electrons, which go and do their thing, and then you have your O2, which is released directly. But if you look at the other ones, A, the oxygen atoms in plant sugars are derived from water. That's not true. They're derived from CO2. And the oxygen atoms in the CO2 produced by plant respiration. There's no talk about respiration. This is about photosynthesis, this question. And then let's look at C. The oxygen atoms in the labeled CO2 were incorporated into plant sugars. Only that's not true. It's not just plant sugars. And then E, the oxygen in the labeled CO2 was released as O2. That's not true. The released O2, all of it comes from splitting water. CO2, that O2 goes elsewhere, like in organic compounds. So yeah, D. All right, take a look at this one. Okay, so we need to select two answers that are acidic. So let's look at all of these answers. The thylakoid lumen, is it acidic? Yes, it is, because if you think about that proton gradient we're setting up, we have a lot of protons in the thylakoid lumen and very little of them out in the stroma. So that kind of addresses A and B. A is going to be acidic. If you remember, it's about like a 5, pH equals 5. Chloroplast stroma is going to be about pH equals 8, and it's that gradient that helps you make ATP through ATP synthase. Similarly, the cell cytosol, this is just something you have to remember, but it is a little acidic. And then the mitochondria matrix, it's not acidic because it's kind of like the flip-flop situation of a chloroplast. ATP synthase is pointing into the matrix. So the matrix is going to have fewer hydrogen ions, fewer protons, while the intermembrane space is going to have a lot of and therefore a lower pH. So yeah, that tells us that our answer is A and C. All right, take a look at this one. Okay, so this one's quite nice. You basically just which one is not a plant. Box, it's made from trees, those are plants. Oil, especially like plant oils, think like olive oil, vegetable oil, right? Those come from plants. Mushrooms are not plants notably, they are fungi. And onions, plants, sauce come from tomatoes, plants. So yeah, it's C. All right, go ahead and pause and take a look at this one. Okay, so hopefully you did it. Which of the following does not take part in the light reactions? Okay, so let's think about the light reactions. We have our two photosystems, so it's not A or B. We have ATP synthase at the end, and that's what helps us, use that proton gradient to make ATP. So it's not D. And then the cytochrome complex, it's that middle part of the electron transport chain between the two photosystems. We have plastoquinone, and then we have our cytochrome complex, and then we have plastocyanin. And then we go to photosystem 1. So cytochrome complex, part of that electron transport chain, thus part of the light reactions. So that tells us the answer is rubisco. Rubisco, it's part of the Calvin cycle. It's the enzyme that adds CO2 to RuBP in that first carbon fixation step. And yeah, it's the most plentiful protein in the world. So, see? All right, take a second to look at this question. Okay, hopefully you looked at it. So here we're kind of looking at like a cycle, and then we don't really know where to start. So let's just pick a random starting point, and then let's use that to build an order of these steps, and then we'll figure out which one we should be starting with. So I'm going to start with number 3 because that just feels most logical to me. PEP carboxylase adds CO2 to PEP to produce oxaloacetate. So what happens next is oxaloacetate gets turned into malate or other organic acids, and those are moved to the bundle sheath cells. And that sounds like number 2. Now, the next step is that CO2 is released again in the bundle sheath cells, and then the other thing made is pyruvate. So that looks like number 5. And then next thing is CO2 is put into the carbon cycle in the bundle sheath cells. So that's number 4. And then finally we have number 1 where we're regenerating PEP. Okay, so what this tells me is that the answer is either A or D because they have that same order. However, we're either starting with the mesophyll cells exporting malate or PEP being regenerated. And logically, it would make more sense to start at PEP being regenerated because you couldn't get the malate without having the oxaloacetate from the PEP first. So we're going to go with the answer being D. But yeah, if you noticed, A and D, they're the same order. So it can be kind of tricky, but just go by your logic, like what has to happen first, right? Because in the order of A, it doesn't really make sense that you would export malate first and then, be making oxaloacetate at the end. So, yeah. All right, that's it for today. Thank you so much for watching this video. If it helped, please leave a comment or a like, and subscribe for more content. Thank you so much.